for decades, if you had a certain type of seizure, neurologists would diagnose you with an idiopathic epilepsy.
Right.
Which is really just a very expensive Greek way of saying, your brain looks perfectly normal, and we have absolutely no idea what is wrong with you.
Yeah, it was the ultimate medical shrug, honestly.
I can imagine.
As a clinical neurologist, handing a patient an idiopathic diagnosis was just incredibly frustrating. You run the imaging, the MRI comes back perfectly clean, the patient's intellect is completely intact.
But they're still having seizures.
Exactly. The brain's electrical system is periodically just surging out of control. We couldn't explain why the hardware looked totally fine, but the software was essentially crashing.
Well, welcome to this deep dive where we are taking that medical shrug and completely tearing it apart.
We definitely are.
We are unpacking a pivotal 2018 report from the journal Epilepsia by Mullen and colleagues. And our mission today for you, our listener, is to really explore how that mysterious idiopathic label got stripped away.
Stripped away and replaced by a much more powerful, but honestly vastly more complicated word.
Genetic.
Right.
And I am approaching this through the lens of a medical researcher. So I'm geeking out over the genomic data, the molecular mechanisms, and, the population statistics that crack the code here.
And I'll be bringing this all back to the reality of the clinic. Like how these genetic discoveries actually change the landscape for the real patients sitting right across from me.
Oh, it's huge.
Because calling these conditions genetic generalized epilepsies or GGEs, it doesn't mean what most people assume it means. Mm-hmm. It is very rarely a straightforward inheritance from parent to child.
Okay, let's define the battleground first, though. We aren't talking about some super rare fringe condition here.
Right at all.
GGEs account for a massive chunk of patients. It's around a quarter of all epilepsies, and clinically we are looking at a very specific electrical signature.
Yeah, the generalized spike and wave discharges.
Right, on an EEG, firing at over 2.5 hertz.
And that electrical glitch, it manifests physically in three main ways. First, we see typical absent seizures, which are these brief losses of awareness.
It's like staring spells.
Exactly. The person might just stare blankly for, say, 10 seconds, maybe a slight flutter of the eyelids, but they don't lose muscle tone or fall over.
Okay.
Then there are myoclonic seizures, which are sudden shock-like muscle jerks, usually in the arms or the shoulders.
Just a quick jerk.
Right. And finally, the generalized tonic-clonic seizures. This is the classic full-body convulsion that most people picture when they think of the word epilepsy.
Okay, let's unpack this. Because if the hardware of the brain looks completely flawless on a scan, researchers had to prove, beyond a shadow of a doubt, that this software glitch was actually hardwired into the DNA.
And that's where the twin studies come in.
Exactly. The proof in this report comes from identical twin studies, and the numbers are just staggering. Monozygotic twins share 100% of their DNA, right?
Right.
Well, if one twin has a GGE, there is a 76% chance the other twin will develop it as well.
Which is a massive number. To really understand the weight of that, you have to compare it to fraternal twins, who share about half their DNA, just like standard siblings do.
And what's their rate?
Their concordance rate plummets to just 33%.
Wow. That's a huge drop.
It is. For further context, if you look at a highly complex disease like multiple sclerosis, identical twins only have a 30% concordance rate.
Oh, wow.
Yeah, so that 76% figure for GGE, it screams that the genetic code is the primary driver of the electrical instability.
But here is where my genomic data hits a total brick wall in your clinic.
How so?
Well, if it's so aggressively genetic, you'd think it would follow basic Mendelian inheritance rules, like we all learned in high school biology.
Right, the Punnett squares.
Exactly. You expect a 25% or 50% risk for offspring, depending on whether a trait is recessive or dominant. Yet the report states the standard siblings of GGE patients only have about an 8% risk of developing the condition.
It does seem contradictory.
It feels like a mathematical impossibility. How can a disease be undeniably genetic in identical twins, but barely register in standard family trees?
Well, it requires a complete shift in how we think about inheritance. We are conditioned to think of genetic diseases as a single broken switch, like cystic fibrosis or Huntington's.
You either have the broken gene or you don't.
Precisely, yeah. But GGE is fundamentally different. The vast majority of cases operate on polygenic inheritance.
Meaning many genes.
Yeah, we are dealing with a complex interplay of multiple highly common genetic variants that are just circulating quietly throughout the general population.
Okay, so it's not a single broken genetic switch. It's more like a complex recipe.
I like that.
Imagine you're trying to bake a very specific, unwanted epilepsy cake. You might inherit the flour and the sugar from one side of your family.
But those ingredients alone are totally harmless.
Right, they don't make a cake. Unless you randomly inherit the exact right proportion of baking powder and eggs from the other side of the family, the cake never bakes.
That's a great analogy.
So these individual traits just float around homelessly for generations until they perfectly align in one specific individual's DNA.
That visual captures the mechanism beautifully. The traits are silent until the threshold is crossed. And the clinical implication of this is really profound.
Oh, so.
It's the exact reason why the International League Against Epilepsy still permits the use of the older term idiopathic.
Wait, really? Even though we know it's genetic?
Yeah, because telling a parent a disease is genetic carries immense cultural weight. They often internalize this unearned guilt, assuming they pass a single broken gene to their child.
Oh, wow. I never thought about that.
It's heartbreaking. So explaining the polygenic recipe, it helps them understand it's really just an unpredictable genetic lottery. Nobody's at fault.
That makes a lot of sense. But if 99% of these cases require that incredibly complex polygenic recipe to trigger a seizure, my researcher brain immediately zeroes in on the 1% exception.
Always looking for the outlier.
Always. I mean, what happens when a single gene does have the sheer power to cause the disease entirely on its own? Here's where it gets really interesting. The report highlights the SLC2A1 gene.
Ah, yes, the grand exception to the rule.
Yep.
For a neurologist, catching a variant in this specific gene is arguably the most critical diagnostic maneuver we can make.
They said so treatable.
Exactly. The severe form of this variant causes classic GLUT1 deficiency syndrome, which is characterized by profound intellectual disability. But the milder form, it actually hides within the GGE spectrum.
It's an imposter.
It really is. It presents as early onset absence epilepsy.
Okay, we need to look at the mechanics here to understand why. So the SLC2A1 gene is responsible for coding a protein called GLUT1. And GLUT1 has one exclusive, incredibly vital job.
It's a transporter.
Right, it is the primary transporter that ferries glucose, so blood sugar across the blood-brain barrier. And the brain is an absolute energy hog. It consumes roughly 20% of the body's glucose.
It's a massive drain on our resources.
So if that gene is mutated, you don't build enough transporters. The fuel pipe is essentially pinched shut and the brain starts to starve.
And that energy starvation dictates the symptoms we see. Typical childhood absence epilepsy usually starts around age 5 or 6.
Okay.
So if I see a child having those staring spell absent seizures before their 4th birthday, that unusually early onset is a massive red flag.
Because the brain is stalling out early.
Exactly, because it's starving. And there's a secondary mechanical symptom tied directly to this energy crisis too. It's called paroxysmal exertional dyskinesia.
Okay, the terminology there is pretty heavy. I mean, we see words like dyskinesia, dystonia, chorea.
Right, it sounds like a lot of medical jargon.
Yeah. So what is actually happening to the patient's body mechanically?
Well, think about a teenager running sprints. The physical exercise drastically increases the brain's overall demand for energy.
Makes sense.
But because the GLUT1 transporter is defective, the brain simply cannot pull in enough glucose to meet that huge spike in demand. So the motor cortex literally runs out of gas and starts misfiring.
Oh, wow. And that causes the movement issues.
Right. This misfiring leads to dystonia, which are these sustained involuntary muscle contractions causing abnormal postures. And it also leads to chorea.
Which comes from the Greek word for dance, right?
Exactly. Meaning unpredictable, jerky, involuntary movements. Their limbs basically cramp and twist simply because the brain is metabolically depleted.
That is wild. But the treatment for this is the most brilliant part of the entire report.
It really is.
We have a profound structural genetic defect at the blood-brain barrier. The obvious 21st century assumption is that we need some billion-dollar CRISPR gene therapy to, rebuild the door.
But we don't.
No, we just change the patient's diet. Wait, so we have this profound genetic defect, and the fix is just changing their fuel source. Are we missing this in clinics?
What's fascinating here is how often it used to be missed, but now we know to look for it. We put them on a strict ketogenic diet, high fat, very low carbohydrate.
Because of the biochemistry. When the body is deprived of carbohydrates, the liver begins breaking down fat into molecules called ketones to use for energy.
Yep.
And here is the magic trick. Ketones do not use the defective GLUT1 transporter to get into the brain. They have their own completely separate doorway across the blood-brain barrier called the monocarboxylate transporter.
Beautiful.
You literally bypass the genetic defect entirely by feeding the brain an alternate fuel. It is a complete metabolic hack.
It is so elegant, and it changes the entire trajectory of the patient's life. By switching the fuel source, we can effectively stop the intractable seizures and actually protect their cognitive development.
But you have to know to look for it.
Exactly. It requires the clinician to actually ask the right questions about, exercise-induced cramping, or to notice that unusually early age of onset.
Yeah.
If we just throw standard pharmaceutical anti-seizure medications at a starving brain,
Is nothing.
it does absolutely nothing to solve the underlying energy crisis.
Wow. Okay, let's pan out from the single gene exceptions and look at the broader structural anomalies hiding in the DNA. The report dives into copy number variants, or CNVs.
Right, moving away from the single genes.
Yeah, we aren't talking about a single mutated gene or a complex recipe of minor traits anymore. We are talking about entire missing chunks of chromosomes.
Large deletions.
Specifically, deletions on chromosome 15, like the 15q13.3 deletion, or on chromosome 16. About 3% of GGE patients carry one of these missing chunks.
And it is critical to differentiate how a CNV functions compared to the single gene GLUT1 defect we just discussed. A CNV is a risk multiplier, not an absolute destiny.
Risk multiplier, got it.
If a person carries that massive 15q13.3 deletion, their actual chance of developing GGE is only about 30%.
Really? That low?
Yeah. I have seen families where a parent and a child share the exact same missing chromosomal chunk. The parent is completely neurotypical, and the child has severe intractable epilepsy.
Let's visualize how that's physically possible. The DNA is the blueprint for the human body. A CNV means pages 40 through 60 of the blueprint are simply torn out and missing.
Right.
You'd assume that would be catastrophic every single time. But the body has backup systems. It attempts to build the brain using the remaining pages.
And sometimes it succeeds.
Exactly. Whether or not the building ultimately collapses, whether they get epilepsy, depends heavily on what other minor polygenic flaws they have hidden in the rest of their unique blueprint.
That blueprint analogy holds up really well clinically. And those missing pages, they don't just put the building at risk for epilepsy.
What else do they do?
Well, those specific chromosomal deletions were historically identified in studies of autism spectrum disorders, intellectual disability, and schizophrenia.
Oh, wow.
Yeah, they destabilize the foundation of neurodevelopment across the board. So if a patient has GGE plus an intellectual disability, the likelihood of finding a CNV skyrockets.
This actually brings up a wild paradox in the genome-wide association studies section of the paper. They looked at a gene called VRK2.
Yes, the VRK2 paradox.
The data shows that a specific variant in this gene pushes a person closer to developing GGE. It actively promotes electrical seizures.
Right.
But simultaneously, that exact same variant chemically protects them against developing schizophrenia.
It's incredible.
How does one single genetic tweak cause a neurological earthquake but prevent a psychiatric one?
Well, we have to look at the mechanics of signal transduction. VRK2 is involved in how cells communicate and regulate themselves at the synapse.
Okay.
And the brain's chemistry is an incredibly delicate seesaw. If a genetic variant slightly alters how an ion channel opens or how a neurotransmitter is cleared, it might make the electrical network hyperexcitable.
Which leads to seizures.
Right. But that exact same state of hyperexcitability might somehow prevent the specific synaptic pruning or dopamine imbalances that lead to schizophrenia.
That's fascinating.
We are seeing the crude behavioral outcomes,
Yeah.
seizures versus psychosis, but we're really only just beginning to decipher the microscopic chemical trade-offs happening at the cellular level.
So these CNVs and variants, it's basically like living on a genetic fault line. It definitely increases your risk of a neurological earthquake, but doesn't tell us which building is going to fall or if one will fall at all.
That is a perfect way to put it. And that uncertainty, it introduces a massive hurdle when diagnosing these patients, because we have to consider the mimics.
Ah, the diagnostic illusions.
Yes. A patient can present with generalized tonic-clonic seizures and the classic 2.5 hertz spike and wave pattern on an EEG. They look identical to a standard polygenic GGE.
But they aren't.
No. Structurally, their brain is hiding a fundamentally different, often much more severe condition.
The report mentions subcortical band heterotopia as a prime culprit here. Mechanically, this is a neuronal migration disorder.
Which happens before they're even born.
Right, during early fetal development, neurons are born deep inside the center of the brain and have to physically travel outward to form the cortex.
Supposed to migrate.
In this condition, which is often due to a mutation in the DCX gene in females, some of those neurons literally get stranded halfway through their journey. They form a thick, abnormal band of gray matter deep inside the white matter.
It's essentially a double cortex.
Exactly.
And those stranded neurons, they form aberrant, miswired electrical circuits. To a standard EEG, the resulting seizures look just like a generalized software glitch.
But it's actually hardware.
Right. If you perform a high-resolution MRI and know precisely what anatomical flaws to look for, you see the hardware is structurally compromised.
And then there are the progressive myoclonic epilepsies, the PMEs.
They are devastating. Take unverricht Lundborg disease.
Which is caused by the cystatin B gene, right?
Yes, it's a recessive disorder that impairs the cell's ability to clear out waste proteins. For the first few years, a teenager might present with morning muscle jerks and occasional convulsions.
So they looked identical to a patient with juvenile myoclonic epilepsy.
Exactly, which is a very standard, highly manageable GGE. A neurologist could treat them with standard anti-seizure medications for a decade, feeling totally confident in the diagnosis.
Until the waste proteins build up to a critical mass inside the neurons.
And the neurons slowly begin to die under the toxic load. The patient develops severe ataxia, which is a progressive loss of voluntary muscle coordination, along with action tremors and steady cognitive decline.
It unmasks itself.
Yes, the disease unmasks itself as a fatal neurodegenerative condition.
Let me push back on the broader implications of this, though, because I look at massive genomic databases for a living.
Okay.
If these mimics can perfectly impersonate GGE on an EEG and early clinical symptoms and fool a trained neurologist for 10 years, how much of our genetic research data is contaminated by misdiagnosed mimics?
Honestly, that is the most pressing, highly debated issue in epilepsy genetics today. We refer to it as the phenotyping bottleneck.
Because the phenotype is just the observable traits.
Exactly. If the clinical phenotyping is wrong, if I misdiagnose a mimic as a GGE, then sending their blood to your research lab actively pollutes your genomic data.
Garbage in, garbage out.
Right. The genome sequencing technology itself is nearly perfect now. The human interpretation of the clinical symptoms is the weak link. The cleaner and more rigorous the clinical diagnosis, the more accurate the genetic correlations will be.
Which brings us perfectly back to the actual humans sitting in the examination room. Let's transition from the abstract genomic databases to the emotional and practical reality of the clinic.
Let's do it.
The report grounds all this dense science by walking through two distinct patient cases from the very beginning of the paper. Let's look at the real-world application here.
Case number 1 is an incredibly common scenario. A mother in her early 30s has a standard, well-controlled GGE.
Oh.
She's had typical absence, seizures, and occasional convulsions since childhood. She brings in her infant son, terrified, asking the inevitable question, is my baby going to inherit my epilepsy?
Based on the twin and family data we discussed, her specific condition is highly likely to be that complex polygenic recipe. You know, the flour, sugar, eggs, and baking powder all have to align perfectly.
Right, so my intervention there is primarily psychological. I explain the population data to her. The risk to her son is only about 8%.
That's pretty low.
It means there is a 92% chance he will never experience a seizure.
But to truly put her mind at ease, do you order a genetic sequencing panel for the infant just to be sure?
Actually, I actively advise against it.
Really? Why?
Because her condition is polygenic. There is no single epilepsy gene to test for. A broad genetic panel would simply return a list of variants of unknown significance.
Oh, I see.
It wouldn't predict his actual risk. It would just hand her a piece of paper full of ambiguous medical jargon to obsess over. Reassurance, grounded in data, is the correct medical intervention there.
Wow. Contrast that emotional reality with case number 2. A father comes to you seeking help for his 6-year-old daughter, and the crucial clinical detail here is that she has been having difficult to control absence seizures since she was 2.5 years old.
The moment I hear onset before age 4, my entire diagnostic approach shifts.
The red flag.
Exactly. This isn't typical childhood absence epilepsy. I immediately start probing the father's own medical history, looking for very specific, unusual symptoms.
And what do you find?
He mentions that during high school track meets, He used to get these severe, prolonged cramping and twisting episodes in his legs that eventually forced him to quit sports entirely.
Okay, to a layperson, that's just severe dehydration or bad shin splints, but to a neurologist,
It's the paroxysmal exertional dyskinesia.
the physical exertion of track drained his brain's glucose, the motor cortex misfired, and it caused the dystonia in his legs.
Yes,
He has the SLC2A1 mutation, and he passed it directly to his daughter.
it is such a visceral moment in the clinic when those puzzle pieces snap together. If we connect this to the bigger picture, spotting this genetic clue means I am immediately ordering a targeted genetic test or a lumbar puncture for that little girl.
Because the result fundamentally alters the course of her life.
Completely.
If she has the GLUT1 deficiency, you don't tweak her pharmaceutical dosages. You implement the metabolic hack.
The ketogenic diet.
Right, you put her on the diet, bypass the broken glucose transporter, fuel her starving brain with ketones, and potentially stop the intractable seizures entirely.
While also safeguarding her cognitive development. It is the absolute pinnacle of precision medicine, but it requires the clinician to realize that the father's leg cramps and the daughter's early onset seizures are the exact same genetic disease wearing two totally different clinical masks.
That's incredible.
And the counseling changes drastically too. Unlike the mother with the 8% polygenic risk, this father has a 50% chance of passing this specific single gene variant to any future children. The underlying genetic architecture dictates everything from the daily treatment to their family planning.
It is just incredible. So, bringing this entire deep dive together, what is the core clinical message for you, the listener, today?
Well, genetic testing for epilepsies is advancing at lightning speed.
But its true power right now isn't in blindly swabbing every single patient who has a seizure, right?
Exactly. It is a targeted weapon, not a wide fishing net. The utility of a genetic test is unlocked by the rigorous clinical examination.
The human element.
Right. We test when the symptoms point to a specific actionable mechanism, like an unusually early onset, suspected GLUT1 deficiency, an exercise-induced movement disorder, or a case paired with an intellectual disability that suggests a missing chunk of a chromosome.
So the physical examination tells us when to interrogate the DNA.
And the DNA tells us how to fight back.
So what does this all mean? I want to leave you with a final thought to really mull over. We just walked through how a profound structural genetic defect, a fundamentally broken glucose store at the blood-brain barrier, can be entirely bypassed without ever touching the DNA.
We just feed the brain a different metabolic fuel, so it uses a different door.
The sheer elegance of that ketogenic solution, it makes you wonder, if we can outsmart a severe genetic epilepsy with a shift in diet, how many other incurable genetic brain disorders might have a simple metabolic backdoor just waiting?